big hello to my wonderful PU2 champions out there. I hope all my dear children are doing great. Well, this video is going to be a lifesaver for all of you who have not prepared for your PU2 midterm examination until this particular date. Well, that's not a very good thing. First of all, let me tell you that as a teacher, and now as a friend, let me tell you how I can save you from any sort of stress, on the day of your examination. My dear children, this video has a list of questions which are repeated most of the times, which you can expect. And whenever I would be showing you a chapter, I will be showing you some unique MCQs out there so that you have an idea that these type of MCQs can come. Okay, and I'll try to explain a bit about everything. Also, when I say I will be giving you a list of questions, one thing I want from you, and that is to practice each of them, not just by reading the answers, okay, but by writing it by yourselves. At least the key points you should write it. Otherwise, my dear children, you will forget it, because there might be so many questions, you will forget it at the end of the day, then this video won't be of any use. Okay, so I hope you're ready for that. And if you think that this video is helping you in any ways, do not forget to hit on the like button and let me know in the comment section that ma'am, yes, it helped, ma'am, yes, I'm confident, whatever you feel so. Okay, and if you have any doubts, please let me know that as well. So principles of inheritance and variation, this is a chapter that you should not be missing, even though it's very lengthy. Okay, because in your final board examination gives you 13 marks. So here also in your midterms, it has pretty much weightage out there. So you cannot tend to miss it out. Okay, so the type of unique MCQs that you can get, first one is dependent on linkage and recombination. So this is a question, 37.2% recombinant Drosophila progeny obtained in a T.H. Morgan's dihybrid experiment with the phenotypes red eye color, normal body, and white eye color, miniature body is due to. So they are asking you this much recombination is due to what reason? So always remember a funda that is for linkage and recombination, that is, if at all the genes are very close to each other, whatever you do, these genes would be going on together. There won't be any chance that a new combination can come up. So you call these genes as what? Tightly linked genes. Okay, which is why the amount of recombination, the chances that it can combine with something else, with some other genes and form some new recombinations would be what? Very less. So always remember, I always tell that this is you and your best friend. There is no chance someone else can come in and reform the group. So there is no recombination out there. Okay, so over here, these are tightly linked. They would be having less space between them. Now, on the other hand, when you say more recombination is there, probably it's because the two genes are loosely linked. They are far away from each other, which is why there are chances that there can be more amount of recombination. So this part of the chromosome can go somewhere else, and the other chromosomes part can come over here, making it a new combination altogether. So that is possible over here. So if I say 37.2% recombination, this is more or less, it is actually more. 37.2% recombination is more, my dear children. So over here you can expect this. The answer would be what? They are loosely linked and they have much space between them. If at all, in the question it was like 1.3% recombination is there. If it's that, my dear children, that means they are very close to each other. They are tightly linked genes. I hope this is clear. So many questions comes around it. Who gave down linkage and recombination? It was T.H. Morgan who put down linkage and recombination. He put down the term. Then there was his student, Alfred Sturtevant. What did he say? He said that basically, since I know the percentage of recombination, I also know that the genes are closely linked. They might be here somewhere. So with that, I will be able to tell the location of the genes. Okay, so gene mapping or chromosome mapping was put down forward by Alfred. Okay, it's Alfred Sturtevant. Remember that as well. So that was a student of T.H. Morgan. These type of questions can come from linkage and recombination. Always remember 50% recombination is the highest amount of recombination. So anything closer to that particular range means high recombination. Now going ahead with the second, chromosome maps or genetic maps were first prepared by, I told you, it was Sturtevant. If at all linkage and recombination was put forward by whom? It was T.H. Morgan. Now next type of questions that you can definitely get, law of independent assortment based question. So in a dihybrid cross in pea plants, Mendel got 9:3:3:1 ratio. What does it denote? So we all should know what does 9:3:3:1 stand for. So basically over here you are crossing round and yellow parents with another round and yellow parents which looks like this. This is F1 progeny, you're crossing the F1 progeny for the F2, and now in F2 you get something like this. How do you get something like this? What does it stand for? So basically I started with round and yellow, and here also it's round and yellow, but in the progenies I am seeing new things. So one is round and yellow, 9 of them would be round and yellow, which is a dominant trait. One would be wrinkled and green. So ma'am, where do you find this from? So this was there in the parental thing. So the parents were round and yellow. Just a second, okay? The parents were round and yellow and wrinkled and green, and they got a progeny which was like this. So the parental trait is there. The recessive one is just one over here. Wrinkled and green is still there, round and yellow is still there, 9 of them. Now you need to find new combination. Here 3 stands for what? Why does it go like that? Okay, fine. 3 stands for what? It stands for round, and this was with yellow, right? Now we make a new combination, round with green, and here wrinkled was with green, right? Now we make a new combination, round and what? Yellow. So this is a thing that you need to remember. But ma'am, how is this possible, and how are you explaining this whole answer? So basically, when is this possible, newer combination possible? When the gametes of this particular parent assort independently. That means they go independently. For example, over here I have round, capital R, small r, capital Y, small y. So when you form the gametes, one among these chromosomes can go into a gamete, so it can be capital R, and one among them can go, so it can be capital Y. Now, does it always go like that? Does capital R always go with capital Y? No. There can be another gamete being formed with capital R and small y. Okay, from this type only one is going, from this type only one is going. So here small y is coming this time. Now on the other hand, small r can also go with capital Y. Similarly, small r can go with small y as well. So over here, what do you get to understand? The alleles of one gene, that is for the structure, and the alleles of another gene, that is for the color, are going to assort independently. They don't say capital R will only go with capital Y, which is why you have different type of gametes being formed, and these different type of gametes comes together to form different combination of organisms as well, which you see over there. So alleles of 2 genes are segregating what independently. This becomes your answer. This is law of independent assortment. So always remember, for dihybrid cross, this is a law that applies, and law of segregation is applied when we say that okay, the gametes which are going to be formed, they can be either of the alleles. Any 2 alleles. The gamete can have either of the 2 alleles. So in that you say law of segregation. That means in a pair of chromosome, only one chromosome is going into a gamete. That is what you mean. So capital R, small r do not go together. Only one guy can come in, right? Even from a pair of alleles. So that is law of segregation. So whenever gametes are formed, the alleles get segregated for a character, and only one allele goes to the the gamete out there. So that is law of segregation. This is law of independent assortment. Always remember that. Okay. So the next question, the pedigree chart is given below. Identify the trait responsible for this inheritance pattern. So this is the pedigree chart, my dear children, and you have to tell what is being followed over here. Is it recessive or dominant? Easy way to find it out. If it is not skipping a generation, if it is skipping generation, then it will be recessive. What do you mean by that, ma'am? So over here you see the parents are not affected, but the children are. Similarly, here the parents are not affected, but the child is. What do you see here? Skipping of generation. If at all the parent was affected here, then again here also. If the parents are affected and the next generation also you see the same disorder to be going through, it would be a dominant disorder. But here you clearly see skipping, which is why it is recessive disorder. Now, ma'am, is autosomal or X-linked disorder? So to find the X-linked disorder, just look for crisscross inheritance. What do you mean by that? Suppose over here it is passed from the father, then the daughter, and then the son. So here the father should have been affected, the daughter should have been affected, and from the daughter the son would be getting that particular disorder. You can clearly see in X-linked recessive disorder. That is a way to identify X-linked disorder. Otherwise also in general also you can find it. So over here, my dear children, here the boy child is affected, girl child is affected. The next generation again the boy child is affected. But if you see this generation, they are almost affected equally. They're almost affected equally. Similarly over here also you see that boy is affected, but anyways you don't see majority of boys being affected, which is why you can clearly say it is what? An autosomal recessive disorder rather than X-linked. Okay, so it will be autosomal recessive trait, recessive because it is skipping generation, and autosomal because, my dear children, you do not see the crisscross inheritance pattern which you mostly see in the case of X-linked inheritance. Done? Clear. Now going ahead to the next question, which of the following is not pair of opposing characteristics that Mendel studied? So this particular chart is very, very important, which is why I included this question. Okay, so you should know all these things. So what are the traits that he studied? Which one was dominant, which one was recessive? You should know that. So which is not a pair, it is pink and white flowers, because it is not pink, it is violet flowers that he studied. Violet was the color which was dominant. The flowers which are violet in color, they are the dominant, and the white flowers basically they are recessive, my dear children. Okay, now how do we remember the other characters? Always remember for seed there are two things, seed color and seed shape. So in seed color there is green and there is yellow. Okay, there is green and yellow. So usually when we have green peas, we have green peas. The green color is what we assume is dominant, right? But it is not. Yellow is the one which is dominant in the case of seed color. And you know the experiment which was done in dihybrid cross. So you will remember that yellow is what? In seed color, it is a dominant one. Whereas green is recessive. Now, if you talk about seed shape, round is dominant and wrinkled one is what? Recessive. So two things for what? The seed associated thing. Now we will come to what? The flower. The one feature is what? The flower color, which is violet and white. I told you violet is dominant, whereas white, which is there, it is recessive. Now the next thing about flower is what? The position of the flower. So it can be axial. For example, from the point where a branch is coming off, now a flower is coming. This is axial position. And terminal position is where, my dear children, at terminal things. At the tip, you will get flowers. So which one is dominant? Axial is the one that is dominant. And terminal is the one which is recessive. So how do you remember it, which is dominant? A comes before T. So A is dominant. Okay, so this is for the flower. Now comes the pod. So pod means the covering of the pea. So you might have seen that pea is inside what? A pod. So pod color is there. Green is the one which is dominant and yellow is the one which is recessive. So you always look for the green color pods, right? When you go to the market. So green is the one which is dominant, opposite to what we saw in the seed color. Okay, apart from that, the pod shape. So pod shape can be full like this or it can be constricted like this. That means it is very dry and things like that. So full and constricted. Done. Apart from that, is there anything else? Height of the plant. That is something that is unique, which is not in two features. So height can be tall or dwarf. So tall is the one which is superior, whereas dwarf is the one which is recessive. That's all. So you need to remember all these features. If at all you do not remember it, I hope I have helped you in some ways. Okay, now going ahead with the dihybrid cross. So this question can also be asked, that is in the F2 generation of Mendelian dihybrid cross, the number of phenotypes and genotypes are. So we know the number of phenotypes. Ma'am, you just told 9:3:3:1. So 9 of them green and yellow, round and yellow. 3 of them round and green. 3 of them wrinkled and yellow, 1 of them wrinkled and green. So these are the 4 phenotypes, 4 physical things that I can clearly see. Now what are the genotypes? So you assume that when you do the dihybrid cross, there are 16 outcomes. So 16 genotypes will be there? No. The genetic makeup, which is unique, which is not, okay, I'm not talking about the repeated number of times. Okay, so if at all RRYY is there over here 5 times, I will not count it 5 times. I'll just count it once. So these are the unique genotypes that we are having. How many of them are there? 9 unique genotypes. Okay, so how do you remember it? Again, the phenotypic ratio, you remember 9:3:3:1, right? So it starts from 9. So you can remember the genotypes which are there, they are also 9 in number. Phenotypes, they are 4. Phenotypes, they are 4. 9:3:3:1. 1 phenotype, 2nd phenotype, 3rd phenotype, 4th phenotype. And now genotype, they are 9 in number. You can remember like that. They would not ask you to write down all the genotypes, that won't be necessary. So at least remember this particular thing and look for what generation they are asking. F1 generation, F2 generation, F1, all of them basically they are round and yellow. The phenotype is round and yellow and the genotype is what? Capital R, small r, capital Y, small y, all of them. Okay, Chalo, that won't be asked generally, but yeah. Now, rediscovery of Mendel's work, these questions are also usually asked when it was rediscovered in the year 1900, my dear children. There were three scientists who were behind this. Who were those three scientists out there? It was De Vries, Correns and Chemark. Please remember the name of these three scientists. De Vries, Correns and Chemark, these three scientists in the year 1900, they rediscovered Mendel's work. Mendel did his work from when to when? That is also a question. He worked for 7 years from 1856 to 1863. Okay, 1856 to 1863. If at all you remember 7 years, that would be enough. And if at all you will remember this, that is also enough. So plus 7 would give you this particular thing. Okay, 1856 plus 7. 1856 plus 7, that's all. Okay? Now, Mendelian disorders, questions like this can be asked on the basis of color blindness. So, color blindness, hemophilia are the only X-linked recessive disorders mentioned in your NCERT. Okay, apart from that, all of the other ones are autosomal recessive. Now, only one dominant disorder is given, that too in the pedigree picture, which is of what? Which is of myotonic dystrophy. It is an autosomal dominant disorder. Rest everything is recessive. Okay, and two of them are X-linked recessive. One is color blindness, another one is hemophilia. Now, when is it possible for a woman to be colorblind? So, my dear children, colorblind, as I said, it is an X-linked recessive disorder. So, for a woman to be colorblind, both the X chromosomes should be affected. For a male to be colorblind, at least one should be affected, because there is no other one to overshadow it. Okay, there is no other X chromosome to overshadow it. So, at least one, if it's there, then also he would be what? Colorblind, which is why it makes them more, it is more dangerous for them to have this particular thing. Okay, they have more chances for getting color blindness. So, over here it is said, when can be a woman be colorblind? So, a woman can be colorblind when she gets one X chromosomes from her father and one X chromosome from her mother. Now, my dear children, you only think. Father can only have this condition, he just have one X chromosome, and that X chromosome has to be like this. That means the father would be definitely what? Colorblind. On the other hand, if we talk about the mother, she can have a condition like this where she can be just a carrier. Why a carrier? Because there is another X chromosome to overshadow this effect. Right? It's a recessive disorder. It can be only appearing in the recessive condition. Except in the case of males, because over here there is no other X chromosome to overshadow the effect. Otherwise, she should be like this. So, either she should be colorblind, or she can be also what, my dear children? She can be a carrier. Now, you have to look for options out there. The father has a normal vision. That cannot be a condition, because the father should be carrying this particular defect out there, so he cannot have normal vision. The father is definitely going to be colorblind. So, the mother has a normal vision. Do you think the mother can have a normal vision? Yes. If she is a carrier, she can have a normal vision. But let's be more specific. So, the father is colorblind and the mother is carrier. This is more specific option. So, please go with this one. I hope it's clear. Done? That's it? Okay. Now, these are the 5 marker questions out there which you should definitely refer to. Ma'am, where are the answers? So, when I will give you the PDF, I'll include the answers and I'll give you. Okay, I have not included the answers right over here, because that would just make me talk a lot, okay, about the answers, and you will feel bored at the end. Because I believe that this particular video is for you to refer at the end and watch the answer, see the answer, read out the answer, things like that. If you think you need an explanation, I will put the link to my playlist out there where I have explained all the questions depending on the chapter. For example, principle of inheritance, all these questions that you see, I have explained it there also. Okay, so if you want an explanation, you can refer to that. If you just want the list of questions, here is the question. If you want the answers within the PDF that you're going to get of the passing package, you will get the answers as well. So please do not be worried about anything. Okay, is that a deal? Fine. Now, what do you have to do when I show you the question? Whenever I show you some questions, you have to try to find the answers by yourself, if at all you can, but you don't have to worry. I have already got your back. But my dear children, one thing that you need to do is that please try to at least write the answers by yourself. I do not need you to write the whole entire thing out there. At least write down the key points so that you don't forget. So when I was a student, when I used to read the answer like this, it goes on like that. You will forget it. I am telling you, I forgot. Remember, just imagine you are reading all the answers, my dear children. How do you think your brain will know what to store, what not to store? So first thing that you need to do is, once you're done with an answer, your concept is ready, write it down. The key points, nothing else. Don't waste too much of your time. You have to do it. If you're not doing it, you will forget it. Okay, because it's not about one chapter. There will be multiple chapters, multiple questions out there. So please do it. So what is phenylketonuria? Talk about it. It's an autosomal recessive disorder. And then what exactly happens in there in phenylketonuria? Enzyme is not being released, phenylalanine hydroxylase, which is why what happens? Phenylalanine is not converted to what? Tyrosine. All this thing happens. If it is not happening, then what's the problem? Phenylalanine, since it is not getting converted, is getting accumulated in our body in the form of phenylpyruvic acid, my dear children. Okay, and when it gets accumulated in that sense, it can cause mental retardation. Okay, and since tyrosine conversion is not happening, the tyrosine is very much important for a formation of a pigment, that is melanin. So melanin is not going to be formed, which is why the people who have phenylketonuria will also have reduced pigmentation in their skin as well as their hair. All these things you should know. Please write it down in the answers. It will be there in the answer sheet whenever I give you. Now, quantitative thalassemia and qualitative sickle cell anemia, the blood disorder, you should give the difference between both of them. So, quantitative one is thalassemia. Okay, over here the genes are there. Okay, the genes would be affected. Since there are many genes, if at all a particular gene is affected, the other genes can contribute to it. Still, hemoglobin would be formed. Okay, still hemoglobin will be formed, but the quantity of it will be reduced. Suppose all the genes are mutated, in that sense, it won't be produced at all. So, it's a quantitative disorder. Sickle cell anemia is a qualitative disorder. Why? Because over here, the beta globin chain, okay, the production of beta globin chain is hampered because, my dear children, there is a mutation happening. GAG is replaced by GUG. There's a point mutation. A is replaced by U, which is why GAG was coding for glutamic acid, but now it is coding for valine. Always remember, A comes before U. So it was A before and then it is replaced by U. Similarly, glutamic acid starts with G. G comes before valine. G comes before V. So glutamic acid was there, now it is valine. It is happening at the position of 6th codon. Remember that. What gene is affected? Beta globin gene, HBB gene is getting affected over here. In the case of thalassemia, both alpha as well as beta globin chains are being affected. So go through it. Remember the chromosome number. Alpha globin genes are present on chromosome number 16. Alpha on chromosome number 16. Beta on chromosome number 11. Okay, so all these things you need to remember, have a study about these disorders. There is one more blood disorder. So in your book, there are a total of 3 blood disorders. 2 of them are autosomal recessive. 1 of them is sex-linked recessive. That is hemophilia. Okay, that is just like color blindness. Now you have to give reasons for all these questions. That is, a simple cut results in non-stop bleeding in hemophilia. Why? Because in hemophilia, my dear children, one particular protein, which is a cascade of proteins in clotting, okay, that is getting affected, which is why the clot is not being formed. And if clotting is not there, don't you think the blood will go on? Yes, it will go on. So that is a thing. Okay, now Turner syndrome affected females are usually sterile. Why? Because in Turner syndrome, they just have one X chromosome. They should have got XX chromosome, but that's not the condition. They just have one X chromosome. Now you call this condition as exo condition. This is known as monosomy. Why? Because only one chromosome is there. One chromosome is lost, which is why this condition is known as monosomy of the X chromosome. Over here, why do you think they are sterile? Because, my dear children, since 2 X chromosomes are not there, their ovaries are rudimentary. They are not properly developed. And if ovaries are not developed, do you think ovum will be formed? No, which is why they will be sterile in nature. They are not fertile. Now, in Morgan's dihybrid cross experiment and Drosophila showed that flies having genes for yellow body and white eyes exhibited less recombination. Why do you think they show less recombination? So you do not have to think a lot. Less recombination we studied in the first part of the chapter. First question, right? Less recombination means they are very close to each other. They are tightly linked. They have less distance between them. That's all. Okay, so you just answer like that. Accumulation of phenylalanine in the body of phenylketonuria affected individuals. What does, okay, why does accumulation happens? Because the enzyme is not produced, which is responsible for conversion of phenylalanine to what? Tyrosine. Okay, what is that enzyme? Phenylalanine hydroxylase. Mention that. Okay, now you have to define aneuploidy. Aneuploidy is what, my dear children? Whenever, suppose whenever a cell has to divide into 2, the chromosome duplicates and the chromosome also divides into 2. But over here, my dear children, what is happening? The chromosome is not dividing properly, which is why some cells will get more, some cell will get less. This is known as aneuploidy. Now, aneuploidy can be in 2 different conditions: trisomy, monosomy. Trisomy means you get an extra chromosome. Monosomy means you lose a chromosome. So, example of monosomy is Turner syndrome. Example of trisomy is Down syndrome. In Down syndrome, 21st chromosome, you get an extra extra copy of it. It is autosomal. Okay, it is an autosomal chromosome. Out of the 23 pairs of chromosome, 22 pairs are autosomal. Only the 23rd pair is what? The sex chromosome. So this is happening in the 21st chromosome, which is a part of autosomes. Please remember that. Remember the symptoms of Down syndromes. They have broad pancreas, they would have mental retardation, short stature, all of it should be known. Now Klinefelter syndrome and Turner syndrome, my dear children, you should know how it is caused. So in Klinefelter syndrome, you get an extra X chromosome. So it should have been just XY, but you get an extra X chromosome. Okay, so you should know in our body, generally, there are 44 autosomes. Okay, 44 autosomes, and the last pair of chromosomes is sex chromosome. That can be XY, that can be XX, or whatever thing is there. But over here you have a condition XXY. So what is the number of chromosome? 44, 45, 46, 47. So if you are asked to write the karyotype, this is the karyotype. This is what you mean. 44 autosomes are there, and then one extra X is there, which is why it is plus XXY. Or you can also write it as 47, XXY condition. Now in the case of Turner syndrome, you can write, usually over here in Turner syndrome, what is happening? 44 autosomes are there, and there is XO condition, my dear children. What is happening? There is XO condition. That means 44 and 45 chromosomes. Apart from that, there is no other chromosome out there. So you can just write 45, XO condition. That would be enough. Okay, you can write like that as well. Or you can write it just like this, 44 autosomes, and then. I prefer this because that is more easy for you to remember. Here, autosomes would be just like that. It's a sex chromosome which is going to be affected. Fine. So Klinefelter syndrome, again, you would be asked the symptoms as well. The symptoms of Klinefelter syndrome is what? So they would have a masculine body. Okay, but again, they would have gynecomastia. What do you mean by gynecomastia? Development of breast. So in a male body, development of breast. And they are going to be sterile as well. Just like Turner syndrome, here they are also going to be sterile. Because over here, X is overshadowing the effect of Y, my dear children. They would have feminine character as well. Okay, their spermatogenesis is not going to happen. No formation of sperm is going to happen. The secondary sexual characteristics also is not going to be there properly. In Turner syndrome, on the other hand, they will show male characters. They will have masculine body, this and that, and things like that. Okay. Now, incomplete dominance, you have to explain the example of snapdragon. You also call snapdragon as what? You also call it as antirrhinum. And over here, you have to mention that you chose red flowers and white flowers, and when you fused it in F1 generation, you got something like this. But instead of being red, it was pink in color. Why? Because it showed incomplete dominance. And now when you crossed this and found the F2 progeny, you see one very important thing, 1:2:1 ratio in phenotype. This is the phenotypic as well as the genotypic ratio. Phenotypic, why? Because one of them is going to be red, two of them are going to be pink, and one of them is going to be white. Genotypic, why? Because one of them is going to be capital R, one of them is going to be small r, two of them are going to be capital R, small r. So please talk about the incomplete dominance properly and write it down. Now one gene inheritance, in that you have to talk about the Pisum sativa, where you're crossing the tall plant and the dwarf plant. Capital T, small t. F1 generation, what happens? F2 generation, what happens? Everything along with their genotypic as well as phenotypic ratio. I hope that's clear. Okay, now going ahead with what? With one very important thing out there. So my dear children, if at all you are someone who is preparing for your KCET examination, then make sure you are doing your research for this particular course that we are launching, which is KC90, because it is definitely going to help you. We are not just focusing on KCET, we are also focusing on COMEDK, but as a biology student, I think you have scope for KCET, because that's where you can go ahead with BSc nursing as well as agricultural courses. So it's a 90-day crash course, and it is offline, happening at Diksha campuses. So make sure you are going through it once. And if at all you have any doubts, you can ask us in the comment section as well. Okay? And we have a slight chance to make it online as well for those children who are not gonna be able to come over here, okay, at Bangalore. So yes, definitely let us know that as well in the comment section. Now the 2-marker questions which you should definitely prepare is this. Write a short note on sex determination in the case of birds. So in the case of birds, they have ZZ, ZW type of sex determination. So please refer to that. This is the only type where the females are going to produce two type of gametes, which is why you say that this is what? Female heterogamety. So over here they produce Z type of gametes and also W type of gametes, whereas males are going to produce only ZZ type of gametes are there. Only Z out there. Okay, so this is the only case where female heterogamety is shown, and this is in the case of birds. Now, haplodiploid sex determination, where do you find it? You find it in the case of honeybees. What exactly happens over here? If at all it is diploid, that means it has double the number of chromosomes like this. So in them it is 32. Okay, it would be a female. On the other hand, if it is haploid, that means they have half the number of chromosomes. Half of 2n is n, right? So they will be having just 16. In that case, it will turn out to be a male. So depending on what is the number of chromosome, it can be a female as well as a male. Okay, so these things are very much important. This you see in the case of honeybee. Now going ahead with blood grouping. Now blood group is also very important. There are 3 alleles for blood grouping. Okay, one is IA, other is IB, and the other one is IO. Over here, always remember IA and IB are dominant over IO. Okay, so if you get a condition like this, from one parent you get IA, from another parent you get IB, which one will be dominating? Since both of them are dominant over O, are they dominant over, is A dominant over B? Is B dominant over A? The answer is no. They show something that is known as co-dominance. Both of them are equally dominant. Okay, so which is why they are co-dominant, my dear children. Always remember that. So all these facts you should remember, and also this is an example of multiple allelism. You usually studied one allele, capital T, small t. Right, this is the pair of alleles that you study, but over here you have multiple alleles out there. So yes, that is what you see in blood grouping. Now test cross and point mutation is also there. Point mutation, for example, whenever there is a change in the single base pair, for example, I told you it was GAG, but this base is now changed to GUG. This is an example of what? Point mutation. At a single point, a single base pair is getting changed, which is why the whole. So this codes for another amino acid, that is glutamic acid, whereas this codes for what? Valine. So the whole thing, the amino acid is changed. So obviously the amino acid comes together to form the protein as well. So that also will have certain changes out there. So you can give the example of sickle cell anemia for point mutation if at all the question is asked. Now test cross is being asked. What do you mean by test cross? So ma'am, I have a tall plant, but I do not know whether it's tall like this or it is tall like this, because anyway capital T is dominant over small t. So what do? I do test cross. In test cross, I am going to make it fuse with a recessive parent, specifically with a recessive parent. Why? Because in that condition I will get to know things better. So here I have capital T, capital T. On the other hand, I have capital T, small t, small t, small t. Let's see what can be the outcomes. Okay. So here capital T, small t is capital T, small t, capital T, small t, again capital small t, again capital T, small t, again capital small t. All of them are capital T, small t, right? So capital T will dominate, all of them are going to be tall. But in this condition, where I am fusing this one with this recessive parent, so capital T, small t, capital T, small t, and then small t, small t, small t, small t. So two of them are going to be dwarf, two of them are going to be tall. With that I get to understand that this is not the tall I was looking for. This is heterozygous tall, and this one is what? Homozygous tall. Why do you need it? Because if I do crop breeding program, I want a tall variety of plant, I would obviously take a pure breed like this. Pure breed means they are going to be homozygous, they are going to be same. Okay, and if it is not pure, it will be heterozygous like this. Both the alleles would be different. So always remember this. So when you go back and cross, this is also known as back crossing. But when you're specifically crossing with the recessive parent and not the dominant parent, you call this as test cross. Works? Done? Deal? So these are the two markers that you should be referring from this particular chapter, that is principles of inheritance and variation. Now going ahead with molecular basis of inheritance, my dear children, the first type of question that you may get, the number of nucleotide pairs present in the DNA of the primary oocyte of a newborn in human is. So they are asking you what is the number of nucleotide pairs in the primary oocyte. So you believe the primary oocyte has undergone meiosis. No. The primary oocyte which is there, it is diploid in nature, it is still undergoing meiosis. Okay, it is going to undergo meiosis to give you what? The secondary oocyte. So over here it is diploid and then it becomes haploid. So what is the diploid number of nucleotides over here? 6.6 into 10 to the raised to power 9. If it is haploid, half, then it would be 3.3 into 10 to the raised to the power 9. Always remember this. So primary oocyte is diploid, not haploid, which is why it would be 6.6 into 10 to the raised to the power 9. Sorry, this is the answer. Okay, fine. Going ahead, the DNA segment has a total of 1000 nucleotides. 1000 nucleotides are there, out of which 240 of them are adenine. So we know adenine are how much? Adenine is 240 of them. What you have to find out? You have to find the number of pyrimidines. What are pyrimidines? Thymine as well as cytosine. How can you find it? So you should know the purine. So purine is adenine as well as guanine. I hope you know that adenine base pairs with thymine. How many times? Twice. Okay, so you can remember 2. Okay, T for 2, and here 2 bonds are there. What bonds? Hydrogen bonds. G and C, because they look very similar to each other, they are very closely bonded, they have 3 bonds out there. Okay, you can remember like that. So over here, if 240 adenine is there, obviously it would be 240 thymine as well. Agreed? Now both of them, 240 plus 240 is equals to what, my dear children? It would be 480. It would be 480. Right, total of. So out of 1000, 480 of them are adenine as well as what? Thymine. So 1000 minus 480 is equals to what? Is equals to, gives me 520. Now 520 I have to divide between 2 of them because 2 other nucleotides are there. So 520 divided by 2 gives me what? 260. So this is 260 and this is also 260. So ma'am, my answer would be what? Your answer would be this plus this, because you're asked about pyrimidines. So you have to plus thymine as well as cytosine. So thymine is 240 out, and then 260 is what? Cytosine. So basically you just have to plus it. 240 plus 260 gives you what? 500. So this becomes your answer. Okay, so these type of questions can also come in. Now next thing, GGU, GGC, GGA, GGG all code for glycine. Which salient feature of the genetic code has to be proved? So what are the features of the genetic code? This can be a 2-marker question as well, so 3-marker question as well. So be prepared for all this thing. Okay, so here it is said this all code for glycine. What feature does it show? It shows degenerate feature. This feature is known as degenerate, where more than one codon can code for the same amino acid. Now if at all I was saying that GGU is only coding for glycine, what feature is it showing? It shows unambiguous nature. That means one codon does not code for any other things, amino acid. It just code for what? Glycine, one amino acid itself. So one codon codes for only one amino acid. Now always remember they are triplet. Okay, they are triplet. The start codon has the function to start the process of translation, and also it codes for an amino acid that is known as methionine. What is the code? It is AUG, AUG. Okay, now 3 stop codons are there: UA, UGA, UAG. Okay, UA, UAG and UGA. So these things are there. They are stop codons. They do not code for anything out there. And always remember the codons which are there, they are commaless. Now if I have a question that ma'am, GGU is coding here for glycine, and in E. coli also it's coding for glycine. What does it show? It shows that it is universal. That means the codons are going to code for the same amino acid in my body and also in the bacteria's body. It shows it is universal. So over here that is not being asked, but I was just explaining you all these points. Make sure you write it if at all genetic code is being asked. Okay, so this feature is known as degenerate codons. Now going ahead to the next type of question: if a processed mRNA of a particular cistron contains 636 base pairs that code for a polypeptide chain, what are the number of amino acids that it codes for? So 636 bases are there, my dear children, and you have to find the number of amino acids. So 636 divided by 3, because the base pairs arrange into codons. How many? If we get to know how many codons, we can get to know how many amino acids as well. Okay, so 636 divided by 3 gives me what? 212. These many codons are there, so these many amino acids should also be there. Do you think so? Yes ma'am. No. Please remember there will be a stop codon as well. So you have to remove one codon out of it. You have to remove one. So it will be 211. Stop codon is also there in a cistron. Cistron is what? A unit which has start codon, which has everything, as well as a stop codon. So start codon anyway is coding for methionine. The stop codon does not code for anything, which is why you need to minus that. Okay, so it will be 211. Here is the explanation of it if at all you need it. Okay. Similarly, you will get the explanation of all the questions. Now, in pea plants, each character is governed by a gene which is what? Monocistron, polycistron. Do you know what this means? First of all, understand. So, monocistron means over here, you just have this particular thing, a gene coding for one protein out there. A gene that codes for one protein. Polycistron means 1, 2, 3. Multiple genes are there, and multiple genes are going to code for what? Multiple proteins. First protein, second protein, third protein out there. This is known as polycistronic genes. Okay, this is known as polycistronic. So when do you find this? You find this in eukaryotes. That means they have membrane bound organelles, they have nucleus, proper thing, everything is proper in eukaryotes. And prokaryotes such as bacteria, they have polycystonic thing. So over here plant is what? A eukaryote. Apart from bacteria, all of them are what, my dear children? Eukaryotes. Always remember that. And they would be having monocystron. They would be monocystonic in nature. Cool? Done? Cool. Let's go. Now, match the following. I put this question, why? Because I needed you to remember all the names. So, first of all, I know a simple thing, that is Griffith was the one who did the transforming principle experiment. So, D goes with 1, and with that I can find the answer. There's only one thing that goes up there, but you need to know about everyone. So, Jacob and Monod were the people who found the lac operon. Okay, Jacob, Monod were the people who found lac operon. You can remember both of them has O in there, so lac operon. Okay, now F. Sanger, S for sequencing. So he was the one who went with annotated DNA sequencing technique. Alec Jeffrey has 2 F in it. He was the one who found DNA fingerprinting. Alec Jeffrey for DNA fingerprinting, F. Sanger for what? DNA sequencing, Jacob and Monod for lac operon, and Griffith for transforming principle experiment. Done? Done. Cool. Now here we have the 5 marker question list out there. So list of the salient features of the double helix model of DNA. You have to talk about the DNA. What is made up of? It is made up of pentose sugar, ma'am. Okay, so 1, 2, 3, 4 and 5th carbon is outside. This is pentose sugar, and now on the first carbon nitrogenous base is attached. On the 5th carbon phosphate group is attached. This forms what? A nucleotide. This forms a nucleotide. What is this bond known as between nitrogenous base as well as the first carbon? This is known as N-glycosidic linkage. Okay, what do you call a bond between this 5th carbon as well as phosphate? This is known as phosphoester bond. Okay, this is known as phosphoester bond. But ma'am, in my question was there where another, okay, just a second, okay, where another sugar is there, and over here the 3rd carbon of this particular sugar is also bonding with the phosphate group. Okay, since here also carbon and phosphate is getting bonded, here also carbon and phosphate is getting bonded, so this is also phosphoester bond. And now what do you call this whole bond as? Phosphodiester. Since it is happening twice, it will be known as phosphodiester bond. If at all you are asked the linkage between 3 and the 5th carbon of 2 other nucleotides, it is phosphodiester bond. And always remember, you do not see any oxygen being here, but also an oxygen is there in between the carbon as well as the phosphate group, here and here. Okay, done? Deal? So yeah, so the if suppose over here there is no other nucleotide bonding, so this 3' end is free, so I will call this 3' end. And on the other hand, if the 5' end is free, I will call this 5'. Okay, so they are going to run parallelly, anti-parallelly. So one will be in 5' to 3' direction, other one will be in 3' to 5' direction. Why? Because over here, suppose I have the nitrogen bases like this. If I put them in 5 to 3' direction again, my dear children, the nitrogenous bases would be outside. I want them to be inside, since both of them should be linked with each other. Okay, so they run anti-parallelly, always remember. And this thing is also very important. This whole thing, a whole turn is known as 1 helix, and 1 helix has 10 base pairs. How many base pairs are there? 10 base pairs. And if you talk about Armstrong, the length of it is 34 Armstrong. If you talk in between how much space is there, it is 34 divided by 10. It would be what? 3.4 Armstrong. Now if you're talking about nanometer, then my dear children, always remember over here in nanometers, it is 3.4 nanometer in length as a whole. 3.4 nanometer in length as a whole. In between, if you are asking, then I will say it is 0.34 nanometer. Okay, so this is known as a pitch. Fine, going ahead. All these things you need to write, okay, and the hydrogen bonds also. How many hydrogen bonds are there between adenine and thymine? 2. T for 2, okay, and guanine and cytosine? 3. Okay, now transcription, rho and sigma factor. Translation, amino acylation, all this portion are important. So transcription as a whole is important, but in that rho and sigma factor is mostly important. So what is rho factor? So for transcription to start, an RNA polymerase have to come and attach on the promoter region. Right, it has to come and attach on the promoter region, and that's when the transcription is going to begin. So it cannot come and attach alone. It needs sigma factor out there. It needs what factor? Sigma. It says that I am sigma, come with me. Okay, and then to end, and then later on, after beginning the transcription process, the sigma factor goes off. The RNA polymerase will do it alone. Now at the end, it wants just to go away. It made the RNA, now it just wants to go away. So it is crying. So rho factor comes in to save it. Okay, so rho factor comes at the end. First thing that comes in is sigma factor. You can remember like that. Okay, now you would be asked to make the transcription unit. Always remember, a transcription unit looks like this. This would be the template strand, this would be the coding strand. Template strand is the one that is going to undergo transcription. So it has to be in 3- to 5- direction. The other one will be in 5- to 3- direction. There will be a region which is known as promoter region. There will be a region which is known as terminator region. Okay, and in between this you have everything out there. The new strand which is going to be formed, it is going to be formed from 5- to 3- direction. DNA polymerase, RNA polymerase, all of them are going to make a new strand from 5- to 3-. Always remember that. Now translation also, you should know what is, you should know all the steps, but amino acylation, what do you mean by that? So for translation to happen, there is an mRNA. I hope you know that this mRNA has information as in codons like A, U, G and whatever thing is there. Now this has to be read by tRNA. So tRNA which is there, my dear children, it needs to be attached to one amino acid. This process is known as amino acylation. What do you call this? Amino acylation. So tRNA, it's an RNA which is having 5- and 3- end. So amino acid attaches at the 3- end. Just give me a second. Amino acid which is there, it attaches at the 3- end. Always remember that. And there will be an anticodon loop over here. So suppose this comes over here. Anticodon A would be what? U. For U it will be A. For G it will be C, and over here I have the amino acid which is known as methionine. So this process where the amino acid is attaching to the tRNA, this is known as charging of tRNA and also amino acylation, and it needs ATP. Always remember that. Works? Cool? Fine. Now explain the working of, other than that you have to explain all the steps. First the smaller subunit comes and attaches, then this particular tRNA will come and attach, then the whole thing would be coming in. So again, explanation, I have explained it previously, but you have to look forward to it. Now explain the working of lac operon. You have to talk about the whole lac operon, what is the role of the regulator out there, what does it do, every single thing has to be explained. List the features of the human genome. Okay, so you have to talk about 3164 this million number of things are there, 30,000 genes are there. All the features which are there of human genome are really, really important. Now experiments, semi-conservative mode of replication done by Michelson and Stahl. Please go through that experiment. DNA is the genetic material. 3 experiments are there. One is of Griffith, then is of Avery, MacLeod and McCarthy, and then it's of Hershey. Please go through the experiment. In Hershey's experiment, bacteriophage is being used. Bacteriophage is a virus. Always remember, it's not a bacteria, it's a virus which affects the bacteria. Okay, so all these things has to be covered, my dear children. Make sure you're going through it. Now going ahead with the next thing out there. Okay, the 2-marker question. Which sequence of bases transcribed from DNA are found both in hnRNA and mRNA? hnRNA is heterogeneous nuclear RNA. mRNA is the processed mature RNA. So basically, whenever I say transcription is happening from the DNA, I get an RNA. This RNA will be having regions that are going to code for proteins. This RNA are also going to have regions which do not code for anything. I call these regions as introns. They do not code for anything. And I have to remove it. Then only I can make a proper mRNA. So this RNA which is being found is known as hnRNA, and I do processing. What is this processing known as? Splicing, where I remove the introns. There is one more processing where I put cap at the end. I add some chemical groups at the end over here. Okay, and then at the tail region also I add so many adenine, that is known as tailing. After capping, tailing and splicing, you get the final mRNA which is known as mRNA. So if you are asked what's common here and here, it is the exons. Exons will be carried forward, introns would be removed. Okay, now mention the levels where gene expression can be regulated in the case of eukaryotes. So in the case of eukaryotes, my dear children, first, I can let the transcription happen. I cannot let it happen. The first thing is that. Second thing, I can, I can let the processing happen. I can stop the processing to happen. That is there. Third thing, I can let the mRNA, the processed mRNA to go out. I can stop it. Okay. And the fourth thing is that I can let translation to happen or I can stop it. So these are the 4 ways where I can regulate the gene expression in the case of eukaryotes. In the case of prokaryotes, they do not have a regulation like this. Why? Because over there, in their case, everything is happening in the cytoplasm only. They do not have a nucleus and things like that. So in our case, transcription is happening within the nucleus. mRNA is formed within the nucleus, and then later this mRNA moves out of the nucleus. You get it? So we can have regulation at so many levels. Now euchromatin and heterochromatin, my dear children, that is also very, very important. So make sure you go to that. What is euchromatin? So so we have the histone proteins, 8 of them. You call this histone octamer, right? Now around it you find the DNA wrapping around twice. Okay, why does the DNA wrap around twice? DNA is negatively charged, whereas the histone proteins have positively charged amino acid lysine and arginine. Lysine and arginine. Okay, and now my dear children, suppose the histone proteins are like this. They are far away from each other. That means you have so much space. Whenever, and now this whole thing is known as chromatin. This whole thing, which looks like bead on a string up thing, it is known as chromatin. When chromatin condenses, you get chromosome. So this type of chromatin which has so much space between them, it is known as euchromatin. And since it has so much space, here RNA polymerase can come and do transcription also. So it is transcriptionally active. And since it is far away from each other, they stain less. So they are going to stain lightly, and they are loosely packed, and my dear children, they are transcriptionally active. These are the features of euchromatin. Now make it ulta for heterochromatin. If at all they are very close to each other, do you think it will be transcriptionally active? No. Do you think it will be lightly stained? No, it will be darkly stained. Do you think it's loosely packed? No, it is tightly packed. That is for heterochromatin. Okay, now criteria for a molecule to be a genetic material. What are the criteria? It should be structurally very stable, chemically and structurally stable. It should be able to replicate, otherwise how can I make copies of it? And third thing, it should be able to do slow changes. I'm not saying fast changes. If it changes very fast, my dear children, we won't be like this. So slow changes will be there, should be there. Why? Because we need to have evolution happening. Good things should be coming in. Now the fourth thing is that basically it should show Mendelian characteristics, as in the genes which are there, they should be coding for proteins and everything are there. So four things, always remember. First is replication. Second one is it should be chemically and structurally stable. So which is why RNA was replaced by DNA. RNA was not stable. Why? Because in the case of RNA, they had the same pentose sugar, but in the second carbon, they had OH group, which made it highly unstable. So it should be having some changes, but not so much changes. So that it changes the whole thing, right? So it should be chemically and structurally stable. The third thing, it should be allowing slow changes for evolution. And the fourth thing, it should be able to show the Mendelian traits. That's all. Cool. And now we come to the next thing, that is sexual reproduction in the case of flowering plants. And what do we have? So over here again we have certain MCQs out there. Select the correct sequence of anther walls, anther wall layers. So you have to talk about the anther wall. First is epidermis, then comes endothecium, then comes middle layer, and then comes tapetum. You would be asked to make the diagram of this as well, of the anther. So make sure you're going through it. Now select the correct sequence of events in microsporogenesis. First of all, you have microspore mother cell. My dear children, this will undergo meiosis, and at the end, okay, meiosis happens in two stages. At the end you will get microspore tetrad. This microspore tetrad, they will move away, form single microspores, which later forms the pollen grains. So sporogenous tissue, from the sporogenous tissue which is there, each of them cells which are there, they are microspore mother cell, and they will form microspore tetrad and then later microspores. So your answer is A. You should know the flow of things. Now function of filiform apparatus is 2. Where do you find filiform apparatus, my dear children? You know there is embryo sac, and in embryo sac there is synergid, and then there is egg cell, that is a female gamete out there. So this synergids have a structure that is known as filiform apparatus, which is going to guide the pollen grain. Say that, hey pollen grain, you have to come through this, so that you can meet the egg cell out there, because the pollen tube is going to carry what? It is going to carry the male gametes. So to guide the entry of pollen tube, you have filiform apparatus in the synergids. And always remember, when the pollen tube is going to go, the one synergid is going to degenerate also. Now, from the statements given below, choose the option that is true for a typical female gametophyte of a flowering plant. What is a female gametophyte? The one that will form the gametes. It is what? It is the embryo sac. So you should know what is true for the embryo sac. It is 8 nucleate and 7 cell at maturity. Yes, it has 8 nucleuses. Okay, so if at all this is an, okay, I have the picture of it. So if you see 1, 2, 3 cells, 1, 2, 3, again 3 plus 3, 6, and this whole thing is what? A cell. 7 cells are there. But if we talk about nucleus, 1, 2, 3, 4, 5, 6, 7, 8. So one cell is having 2 nucleus. So it is why it is 8 nucleate, but 7 cell at maturity. Now it is free nucleate during the development. That means whenever the nuclear division takes place, my dear children, it is not covered by the cell wall immediately. First of all, the nucleus divides. How many times? Thrice. So there are 3 mitotic division that happens. That is how you get 8 nucleuses at the end. 3 mitotic division that happens. That is how you get 8 nucleuses at the end, and later on cell wall formation takes place. So A and B, both of these statements are true. Okay, now it is situated inside the integument but outside the nucleus. It is wrong. Everything is happening. This is the ovule, suppose. In the ovule, you would be having nucleus. Inside the nucleus, this particular thing is there. Outside the ovule, you have integument. So integument, then the ovule. Inside the ovule, you have nucleus and the embryo sac. So there is no outside the nucleus. It would be wrong statement. Now it has an egg apparatus situated at the chalazal end. So over here, this is the egg apparatus, synergids plus the egg. This is what you call as egg apparatus, and it is not at the chalazal end. It is at the micropylar end. Okay, it is at the micropylar end, not the chalazal end. The chalazal end is where antipodal cells are there. Everything important happens in the micropolar end. Always remember that. So your answer would be just A and B, because that is the only thing that is correct. Going ahead, even in the absence of pollinating agents, seed setting is assured in. So you should know the example of clistogamous and chasmogamous flowers. Commelina, oxalis and viola are what? Flowers which show, plants which show clistogamous and chasmogamous type of flowers. So over here, clisto means they are closed. They are closed means they have the female as well as the male part. Okay, female and the male part, both of them are there, my dear children. So this would be what? This would be definitely showing pollination. You do not need any pollinating agents out there. Since they are closed, they have everything out here itself. So Commelina, oxalis as well as viola has clistogamous flowers, and they are found in the root region or in the rhizoid region. Always remember. Or they can be little bit above the soil, but not over here. In the aerial region, you have the chasmogamous, the open flowers. For the open flowers, yes, you need pollinating agents and everything out there. Okay? So Commelina is the answer for this question. Now going ahead with the next question out there. The 5 markers. So over here, these are the 5 marker question that you can expect. Characteristics of flowers pollinated by. So in wind, what are the features that you see? So wind, the pollen grain should be light, it should be non-sticky, it should move around. The stamen which is there, it should be high above. Okay, the stigma and the style which is there, it should be sticky in nature. Right, because it wants everything to be coming close, and also it should be feathery. You can say not sticky, you should rather say it's feathery, so that you can trap all the pollen grains that is coming through the wind. Okay, and my dear children, they would be having only 1 ovule per ovary. Why? Because wind pollination is not necessary to happen. It can happen, it may not happen. So basically, why to waste ovules? So only 1 ovule per thing, and they would have inflorescence as well, such that they can have more stamens, stigma and everything out there, so that it can trap things. Okay, example of wind pollination, always remember corn cob and also grass. Grass shows wind pollination. Now insect pollination, again the flower should be fragrant if it is good pollinating agents, or otherwise if it is honey, not honeybee, housefly or anything else like that, it can be having foul smell, and the pollen grains should be very sticky so that it can stick on the body of the pollinating agent. All these things are very much important. They should give nectar, something should be there, or a place for them to lay the eggs. Okay, so you can give the example of that as well. So all these things are important. Reptiles can also act as pollinating agents. Lemurs can, monkeys can, apes can, all of it can. Okay, so know the names of all of it. And water pollination, you should be telling what are the features out there. There are 2 plants given in your book. One is Zostera, okay, that is known as sea grass, and the other one is I forgot the name of it. Zostera, and the other one was, if at all you remember, put it down in the comment section. But please go through the names of all these things, very, very important. One is surface level, at the surface level it is going to do the pollination, and the other one which is there, Vallisneria, yes, that is the name. So Vallisneria which is there, it is going to pollinate at the surface level in the water itself, but in the case of Zostera, it is going to pollinate within the water itself. Okay, so that is the thing. Now structure of megasporangium, you should know the structure of megasporangium. How does it look? So over here you have the ovary. Inside the ovary you have the ovule. The ovary is going to nourish the ovule with the help of a placenta. The placenta is attached with the help of a funicle. The point of attachment is known as hilum. It has 2 integuments, one integument, inner integument. This one is outer integument. Over here you have a space, this is known as micropyle, my dear children. And inside the ovule you have so many cells. This is known as nuclear cells, my dear children. One cell is going to form the megaspore mother cell, which is going to later divide. All these things are really, really important. The opposite end is known as chalazal end. This end is micropylar end. Okay, so here only the MMC will be dividing at the micropylar end. Here only the egg apparatus is going to be formed. All of it very, very important. Now ploidy of cells in the embryo sac. So the question that we did for the embryo sac, which was it? Yes. So what is the ploidy of cells which are there? So always remember, all of them are haploid. Central cell is, since it has 2 nucleus, it will be diploid. So N plus N, 2N. Okay, now what is a ploidy after fertilization, if at all you're asked? So egg becomes egg and the sperm comes in, the male gamete will be coming in and forming a zygote which is 2n. This two polar nuclei will be fusing with another male gamete out there. So 2n plus one more n, triploid in nature. They will give rise to primary endosperm cell. So since two fertilization happening, you call this as what? You call this as double fertilization as well. So ploidy of the cells after fertilization, before fertilization. So I'll write it. After fertilization as well. Okay, now explain the outbreeding devices. You have to talk about the outbreeding devices. Outbreeding means they do not allow self-pollination to happen. So first method is what? Genetic incompatibility, where the pollen grains land on the stigma but still nothing would happen because genetically they are incompatible. Okay, the second thing is that positioning of anther in the stigma. One, the positioning, if at all it's not correct, my dear children, the anther is low, the stigma at the high position, obviously it won't be happening. So that is one type of outbreeding device. The third type is maturation, different time of maturation. The anther might mature later or the stigma might mature later depending on that. Then production of unisexual flowers, the flowers which just have one sex. Okay, either the male or the female. So obviously you need other plants probably for doing the fertilization. Okay, so in that you have monoecious, dioecious. There is too much of detail, but if at all you want an explanation, that please watch the video. It is going to help you really well. So yes, now for the 3 marker, LS of the grass embryo, that means monocot embryo diagram, you should know, dicot embryo diagram, structure of the anther. So these 3 are the diagrams. Parthenocarpy and false fruit. What do you mean by parthenocarpy? If at all the ovary, so usually ovary develops into a fruit and ovule develops into what? A seed. So if ovary is developing into a fruit without fertilization, my dear children, this is known as parthenocarpy. What is false fruit? When apart from ovary, something else is also participating in the formation of a fruit. For example, apple. In the case of apple, the thalamus, which is a part of a flower, it also comes and become a part of a fruit. Okay, apomixis and polyembryony. So apomixis is when a seed is being produced without fertilization, very rare, and in that condition is polyembryony, where you have multiple embryos within a seed. Okay, multiple embryos within a seed, my dear children. That is polyembryony. You find polyembryony in citrus fruits. Okay, apomixis you see in Asteraceae, grasses and things like that. Now structure of pollen grain, the sporopollen, very important thing. If the structure of pollen grain is asked, what are the cells which is there? So one is big cell, which is known as vegetative cell, other one is generative cell. The generative cell will later divide and form the gametes, the male gametes out there, right? And over here you have an intine is made up of cellulose and pectin, and exine is there, which is not continuous. Okay, it is discontinuous, it is made up of sporopollen, the toughest resistant material out there. It can resist alkali, acids, everything. Okay, so please talk about it. Okay, always remember 60% of these plants, they release the pollen grain in this stage, in the 2-cell stage. 40% which are there, they release in 3-cell stage, 1, 2 and 3. 40% less, 60% more, you can remember that. Now difference between gytenogamy and xenogamy. So over here, if I have a plant, 2 flowers, pollination is happening from here to here within the same plant. This is known as gytenogamy. So even though it's cross pollination, it's not true cross pollination, because genetically anyways the parent is the same plant. It won't be. Genetically it will be just like autogamy. Genetically it will be just like autogamy. Autogamy is where you have a flower, from here to here pollination is happening, self pollination. Okay, that is autogamy. So it's just genetically it's autogamy, but actually it's cross pollination, gytenogamy. Okay, in xenogamy you have a whole different plant from here to here, crisscrossing is happening. You can remember that. So that is true cross pollination, and there you will find genetic variations as well. Okay, now difference between pericarp and perisperm. So when the ovary becomes a fruit, the wall layers of the ovary is known as pericarp. Okay, it is known as pericarp. Perisperm is what? So I told you, in the ovary, the ovule which is there, here basically, okay, just a second. So I told you, this is the embryo sac and this is the ovule and that is the ovary, right? So in the ovule you have the nuclear cells, it usually gets regenerated, but sometime it might persist like that and it provides nourishment to the growing embryo, okay, within the embryo sac. So over here, what do you call this condition as? You call that as perisperm. What do you call this? You call this as perisperm. Okay, the persistent nucleus, and you find it in 2 plants, B square, you can remember like that. Beetroot and also black pepper. Beetroot and black pepper. I hope that's clear. Done? Fine. I'm going super fast. Why? Because I want to make you pass anyways. Black pepper. And I know you wouldn't be watching big, long videos out there. Now, human health and diseases. This is chapter, but for the time being, I'll directly move to human reproduction first, because I have seen that most of you do not have this chapter for your midterms, right? So human reproduction with the first chapter that I've ever taken. And over here, during embryonic development, embryo's heart forms at the end of. So first of all, when someone dies, you check the heartbeat. Similarly, when someone is forming also, first thing that happens is heart formation. So the first month itself, you can find the heartbeat. Okay, so you can remember it. The first month, heartbeat is there. In the second month, you have the limbs and the digits. In the third month, by the end of the third month, you have the external genital organ. Okay, that you can find the sex of the child. Okay, external genital organ would be there. Some of the organ and organ system will also be developed. So by the end of the 3 months, you would be having everything, limbs, as well as the digits, as well as external genital organs and things like that. These first 3 months are known as first trimester. Always remember, in 1 month you have 4 weeks. So 4 into 3 is what? 12 weeks. You call third month as 12 weeks, or basically the first trimester as well. Do not forget that. Okay, now after that, 4th month is skipped in your NCERT, 5th and 6th month is given. In the month of 5th month, in the 5th month, movement of the body is there. The baby tries to move, and also there will be hair on the head. So first movement and the hair on the head, it's doing the styling thing, okay? It is found in the 5th month. And now in the 6th month, the God says that, okay, you have hair on the head, have hair on the body as well. There will be eyelids that are separated, eyelashes will be formed in the 6th month. And by 6th month, again, you have completed the next set of 3 months. So this is known as second trimester. What do you call this? Second trimester. Again, 6 into 4, my dear children. In a month, 4 weeks are there. 6 into 4, 24 weeks can also be said. Okay. So over here, I have explained you everything. I'm just changing this thing to 5th month. So it is wrong. It's just 5th month. Okay. Now, seminal plasma in human males is rich in. Seminal plasma is a fluid which is being released by the accessory glands. Okay. How many glands are there? 3. First is seminal vesicle, second is prostate, the third is bulbourethral. Seminal vesicle release fructose, okay, which is responsible for nourishing the sperms, whereas prostate gland is responsible for calcium and enzyme secretion. These are very much important for the process of fertilization to happen. Bulbourethral gland is just for lubricating the penis. Always remember, these 2 glands are the ones which are paired. Okay, you have a pair of them. Prostate is a single thing. Okay, prostate is single. So seminal plasma, secretion of all of it together, you call this as seminal plasma. Seminal plasma plus the sperm makes it what? The semen. Makes it the semen. So seminal plasma is rich in fructose released by seminal vesicle, and calcium which is released by this guy, that is prostate. So you don't have glucose and anything else, please do not follow that. Okay, so it is fructose. Fine. Now going ahead, you have this thing. Role of hormones in spermatogenesis. This question is also very important. So my dear children, males, basically when they hit puberty, that's when the process of spermatogenesis begins. So first thing is that the hypothalamus, which is there in the brain, it releases gonadotropin releasing hormones, GnRH, gonadotropin releasing hormones. Now that will go and act on the anterior pituitary, and the pituitary releases 2 things. One is LH and the other one is FSH. LH acts on the Leydig cells. Okay, L for Leydig cells. LH acts on the Leydig cells, and the Leydig cells are responsible for production of what? Androgens, the male sex hormones. Okay, the most famous one is testosterone. The most famous one which is there, it is what? Testosterone. Now FSH on the other hand acts on the Sertoli cells. Okay, Sertoli cells. Now Sertoli cells, so this is happening here. So in the seminiferous tubules, you have the Sertoli cells as well as the germ cells out there. And over here, you have the Leydig cells. So LH is going to tell the Leydig cell that you have to produce the androgens, and the androgens now gets inside. Why? Because the germ cells which are there, it is going to form the sperms. It is going to form the sperms. Now the Sertoli cells are there for nourishment of this guy. And when I say nourishment, it's not just only nourishment, other things are also there. So FSH acts on the Sertoli cells such that it releases some factors. Why do you think these factors are there? So that the, the testosterone which has come over here, they stay there. They stay there for the whole process, of spermatogenesis to happen, and especially one very important step to happen, which is known as spermiogenesis. So once spermatogenesis is ended, you get 4 spermatids. Now they are like kids. You want to transform them into mature things. So you say it is you want to transform them into what? Spermatozoa. Okay, and this process is known as spermiogenesis. And for this, you will definitely need a lot of testosterone out there. And for testosterone to be here present, you need some factors which is released by the Sertoli cells. So if at all you are asked, if FSH hormone is not being released, which is the thing that is affected? It is the formation of what? Spermatozoa. So indirectly, the process of spermiogenesis that is getting affected. Cool? Cool. Now, oogenesis versus spermatogenesis. So these type of questions can come. So to produce 2000 sperms and 400 ova, how many spermatogonia and oogonia are required? So you assume, okay, you just assume one spermatogonia can give rise to 4 sperms. 1 give rise to 4. Over here, 1 oogonia give rise to only 1 ovum at the end. So if at all I am saying 2000 sperms are there. So 2000 divided by 4 gives me what? 500. So 500 spermatogonia, each of them gave rise to 4 sperms, which is why I have 2000 sperms. So 500 spermatogonia is there. What about the oogonia? So 400 ova is there. So 400, anyway, 1 oogonium is going to form 1 ovum. So my dear children, it will be anyway 400 oogonia. 400 oogonia is equals to 400 sperms. Sorry, 400 ova out there. So your answer would be B. Okay. So always remember 4 and 1 ratio out there. Now the next question, match the items given in column 1 with those in column 2 and select the correct option given below. So over here you have to talk about the menstrual phase. This menstrual cycle is very, 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 very important. Please remember it. So I'll just give you a gist of it. Listen to me carefully, okay? So the first phase is the menstrual phase, where from the 1st to the 5th day, the endometrial lining is falling down. And at this particular point of time, estrogen and progesterone are very low in amount, which is why this thing is happening. And when both of them are low, okay, estrogen and progesterone released by the ovary, and when both of them are low, 2 other hormones increase, and that is the pituitary hormone FSH and LH. They are going to increase. Now later on, when they increase, my dear children, FSH are going to act on the follicles, and the follicles are going to mature. So now we have entered in the next phase, which is from the 5th day to the 13th day, which is follicular phase, because over here follicles are generating. But you know what? The follicles which are generating, they are going to release some estrogen, which is why the level of estrogen goes up. And if the level of estrogen goes up, the endometrium lining also is going to be formed, which is why it is also known as proliferative phase. The one that shut down is now slowly regenerating itself with the help of estrogen. Now when the level of estrogen goes up really, really high, at this particular point of time, it makes LH surge. Okay, it leads to what? It leads to LH surge. So usually when estrogen is being more, LH and FSH goes down. But when estrogen goes really high at this particular point in time, opposite thing happens. LH and FSH has a peak, especially the LH. And when LH has a peak, my dear children, the follicle which is there, the graphene follicle which is there, it will rupture and it will release what? The secondary oocyte. And whatever is left behind is what you call as corpus luteum. Okay, whatever is left behind is known as corpus luteum. Secondary oocyte is gone, the sperm can come, fertilize, and things can happen like that. And over here, since fertilization can happen, the corpus luteum will be now taking over things, and it will release another hormone which is known as progesterone. And now it will increase. It also releases estrogen, little amount of estrogen, but more amount of progesterone. And since because of the progesterone, the regenerating endometrium regenerates even more better. And it is going to secrete certain things, that is glycogen and things like that. Why? Suppose embryo comes in, it has to provide nourishment, which is why. So it is also known as secretory phase, and it is known as luteal phase. Why? Because corpus luteum is there. But you know what happens? The progesterone, when its amount is increased, it tells the hypothalamus, which tells the anterior pituitary to slow down the secretion of LH and FSH. So LH and FSH goes down, and if it goes down, the corpus luteum was maintained by them. It was maintained specifically by LH. Now it won't be maintained there. So corpus luteum will also die off. And if corpus luteum is not there, progesterone will also go down. Then again the menstrual cycle would be happening. Okay? Please remember. So these are the things I have said it so fast way. I do not know if at all you got it stored in your brain. But as I said, I have given you a clear explanation in the midterm series session. Please watch it. You can just move the cursor and watch the things which you need to understand. Okay, so proliferative phase, my dear children, it is also known as what? Follicular phase. Secretory phase is known as luteal phase, and menstruation is where breakdown of endometrial lining happens. So your C answer is the correct one. Next thing, now the 5-marker questions. Human female reproductive system is important. Make sure you're practicing it. Mammary gland is important. Sectional view of the male reproductive system is important. These are the 3 diagrams that you need to follow. Now, schematic representation of spermatogenesis. What exactly happens over here? So, spermatogonia is there. Then later it matures. Differentiates into primary spermatocyte. So, you have to mention primary spermatocyte. 2N in nature. It undergoes meiosis. You get secondary spermatocyte. Half N, half N. Again, at the end you get spermatids. All of it you have to mention. The spermatids transform into spermatozoa. Please mention it like that. Okay. Now role of hormones in the menstrual cycle. So 2 of them are ovarian hormones, progesterone and estrogen. Talk about them. okay, in each phases. That in the follicular phase, estrogen takes up things and it proliferates the endometrium, then progesterone takes up things in the next phase, all of it. Okay, so actually both progesterone and estrogen together does the things in the secretory phase, but it is majorly progesterone. Fine. And then you have to talk about FSH and LH also, how they help in the ovulation. LH, how does it help in the ovulation to happen? How does FSH help in the follicles to mature? All of it. So talk about that. Yes. Now, parturition is induced by complex neuroendocrine mechanism. So how is parturition? Means expulsion of baby outside the human body, right? So how is it neuronal and endocrinal mechanism? How neurons and endocrine system, both of them are involved. So you have to talk about that. So when the baby is there, when the baby is formed, you know what happens? I'll tell you. So baby is going to release some androgens. Whether it's boy or a girl, it will release the male sex hormone, that is androgens. These androgens are converted into estrogen with the help of the placenta. It is converted into estrogen. Now the estrogen, what it does is that there is a layer that is known as myometrium, muscular layer made up of smooth muscles. The estrogen makes certain receptors active over here. What are these receptors? These are oxytocin receptors. Oxytocin is a hormone and these are the receptors for that hormone. Apart from that, my dear children, the baby which is there, when it tries to move out, there are certain stretch receptors in the cervix. It becomes active and it gives nervous signal. So first nervous signal goes up over here. It gives what? Nervous signal. Array, just a second. It gives nervous signal and it goes up to the pituitary of the mother. In the brain, there is a gland, pituitary gland, and this time it's posterior pituitary, not anterior, posterior pituitary. Now it goes up there, a neuronal signal, and this would be producing, posterior pituitary is going to release a hormone that is known as oxytocin. A hormone is gonna come into play. This hormone comes over here through blood, and now these receptors become active. The myometrium starts to contract. Again this goes down, again the receptors become active, again the signal goes off, again. Okay, so it's a loop that is being created, which is why it is neuroendocrine system. I hope that works. Just a second. Okay. Now, next thing, hormones during pregnancy. So you have placental hormones, you have ovarian hormones, you have hormones which increases several folds in the blood. So you have to talk about the placental hormones. What are they? They are HCG, human chorionic gonadotropin. Okay, and then HPL, please mention about that, and estrogen. There are 4 hormones, only 4 hormones released by the placenta. These are the 4 of them. Ovarian hormones, my dear children, during pregnancy, during the later phase of pregnancy, only 1 hormone is being released, and that is relaxin. It is for relaxation of the pelvic muscles out there so that the baby can easily move out. 1 ovarian during the later phase of pregnancy, always remember it. Hormones which increases several folds in the blood during pregnancy. So that means they are there but increases in levels. They are cortisol, okay, cortisol, thyroxine and prolactin. Remember, all of them are there for the metabolism of the mother, for improving the metabolism of the mother. Prolactin is there for improving the lactation, okay. The mother will soon lactate, right? So it's preparing the body for lactation also. So all of it is very, very important. Fine. Now HCG is the one that is responsible for maintenance of corpus luteum once a lady is pregnant. So you know corpus luteum, as I told you, if LH is not there, corpus luteum won't be maintained. So HCG comes into play and it maintains the corpus luteum so that it can produce enough amount of progesterone so that endometrium lining stays like that only. Okay, so all these things are really, really important. Now comes the next chapter, that is reproductive health, and what are the questions that you can expect. So out of the options given below, choose the correct stage for transfer to the fallopian tube for successful IVF results. Okay, successful IVF results. So over here, my dear children, when you want to transfer into fallopian tube, you do not put an embryo which is more than 8 blastomeres. If you put, it would be leading to ectopic pregnancy. That means pregnancy at a point where we do not, that means pregnancy, the baby will be formed in the fallopian tube itself. It will be a problem for the mother as well as the baby. Okay, so this is known as ectopic pregnancy. So if you're transferring into fallopian tube, it has to be up to 8 blastomeres. Okay, this is known as ZIFT, zygote intrafallopian transfer. So ZIFT can be performed when it's in zygote stage, 1 cell stage, or up to 8 blastomere stage, not beyond that. Always remember. Okay, and if it is 16 blastomeres, you put it in the uterus. You put directly into the uterus. Now next thing, which of the following are not completely curable? There are 3 diseases, all of it with H: hepatitis, herpes and HIV infection. These are STDs which cannot be cured by any ways. Okay, your longevity can happen, life can be prolonged, but it cannot be cured. Hepatitis B, genital herpes and HIV infection. Now, amniocentesis, you know amniocentesis is what? So when the baby is there, my dear children, the baby would be having a fluid around it which is known as amniotic fluid, and from the baby's body certain cells goes into this fluid, and if you take this cell, you can be able to tell the DNA and everything. You will be able to find the chromosomes as well, and with that basically it was used to find the genetic disorders, but people used it for what? Finding the sex of the child, which is why it was banned. Okay, so why is it actually used? What is the purpose of it? It is for detection of the genetic disorders. Genetic disorders like what? That is also important. Jaundice, is this a genetic disorder? No. So you cannot find jaundice with the help of this. Genetic disorders such as sickle cell anemia, Down syndrome, all of it. Okay, fine. Next, the 2-marker question that you can expect is this: mention the 2 medical grounds on which the pregnancies are subject to termination. When it is what? It is risk for the mother's physical as well as the mental health, whereas when for the baby it is risky, the second thing. Okay, also when it is rape or any situation like that, at that particular time also it can happen. It will be better if you talk about the act. Okay, that is medical termination act is there given by the government. If we talk about it, it will be making a lot more sense. What are emergency contraceptives? Something that can be used within 72 hours of unprotected coitus, which can be used within 72 hours of unprotected coitus. It can be oral pills. It can be hormone releasing IUDs. Oral pills, hormone releasing IUDs. All of this can be possible. Okay, why? Because my dear children, they are going to release some hormones like progesterone and estrogen, and this is going to cause an imbalance. Okay, it does not allow the endometrium to be there like that. It does not allow fertilization to happen properly. Okay, so which is why the sperm which is there, it can be in the body for 5 days, but you have to prevent fertilization to happen, and you can do it with the help of emergency contraceptives like oral pills as well as the hormonal IUDs, because both of them has a combination of progesterone and estrogen. I hope that's clear. Now, what are the features of ideal contraceptives? You should know about that. They should not come and interfere within the sexual desire. They should be reversible in nature. It should be cost friendly. It should be economic. It should be easily available. All these points you have to write it down. Now, the popular and the effective contraceptive includes IUDs. Mention the type of IUDs with example of each. Very important question. If at all you are asked what is the most widely accepted contraceptive, it is 2 of them. One is IUD, second one is oral pills. One is IUD, second one is oral pills. Okay, always remember. And which one is the best to space children? It is IUDs. Okay, intrauterine devices, you have 3 of them: non-medicated IUDs such as lipase loop. Then you have medicated IUDs such as progesterone, levonorgestrel and things like that. Okay, and you have copper releasing IUDs such as CU7, copper T and multi-load 375, all of this. Okay, so please remember it like that. Remember the name, remember the examples. Explain ZIFT, zygote intrafallopian transfer. Either zygote or the embryo up to 8 blastomeres can be put inside the fallopian tube. That is known as ZIFT. GIFT is where a mother cannot produce gametes but can provide the space for the baby to grow. So you transfer the gametes to the mother's body. GIFT. Okay, gamete intrafallopian transfer, again in the fallopian tube itself. Artificial insemination, where because of so reason a male is not able to produce probably the sperms are not produced in high amount, or the mortality might be reduced. So in that case, artificial insemination is done. Artificially it is being done. Okay, so these are the things, and you need to go and please watch the video if you're not knowing the concept. Otherwise, you will be at a very bad position, and I do not want you to have that. Now, my dear children, for human health and diseases also, I have included the questions out there. If at all you want, you can just go through it. If at all it's coming for your exam. The lymphoid organs, they are very important. Cancer detection, causative agents, the feature of cancer cells, all of it. Okay, the drugs which are there, especially these drugs which are there, the schematic representation of how does replication work in the case of HIV. They are all the important 5-marker question. Active and passive immunity difference in 3-marker. Allergy, okay, structure of antibody molecule, HIV, how it is HIV transmitted, all of it. So and also malaria. So I have included certain MCQ questions as well. You will get it through the PDF along with the answers, which as I said, I promised you guys. I'm not explaining this because as I seen in the live, not much people are having this particular chapter in their midterms. So I don't want to stress you up with that. So that's all. Okay, and if at all you think that it made sense in some ways, it helped you in some ways, you have a clarity of things, definitely let me know. If you need any help from my end, let me know that as well. How you can get the PDF? You have to join our WhatsApp channel. You can download the PDF from there. Okay, done? Cool. So that's all. Do not forget to hit on the subscribe button if at all you have not done that yet. Wishing you all the best. Bye-bye. Take care.